We have
CRISPR. I
Yeah,
think
we
we
have.
have.
Good morning, everyone. Thank you for joining us. Welcome back. I'm Steve, and we're glad that you're here.
And I'm Scott. And yes, we are. Thank you very much for joining us.
Together, we're Backyard Musings.
Yep. And we're talking about a health topic this morning. In fact, both of our topics today have to do with health, but this is a cool one.
Yeah, we talked about CRISPR before. A new CRISPR, that's an acronym CRISPR, powered light sensor can detect the faintest whispers of cancer in a single drop of blood. Scientists have created an advanced light-based. Scott, I think we talked about something. It was an invention that somebody created where you breathe into it and it could tell if you had any sickness. Do you remember that, Scott? About
Yeah.
a year,
I don't
maybe a year and a half ago.
I don't remember if it was any sickness, but it was something that they were pinpointing something. They could use the water droplets or something in your breath or something like that. Yeah.
Okay, scientists have created an advanced light-based sensor capable of identifying extremely small amounts of cancer biomarkers in blood. The technology could eventually allow doctors to detect early warning signs of cancer and other diseases through a routine blood test. Would you like to know more?
Okay, we like that so far, right?
Yeah.
Biomarkers such as proteins, fragments of DNA, or other molecules can signal whether cancer is present, how it is progressing, or a person's level of risk. The challenge is that at the earliest stages of disease, these molecules exist in very tiny amounts, making them difficult to measure.
Quote, our sensor combines nanostructures made of DNA with quantum dots, love quantum, and CRISPR gene editing technology to detect faint biomarker signals using a light-based approach known as second harmonic generation, SHG, said research leader Han Zhang from the Shenzhen University of China. For once, I'm happy to see this coming from China. I mean, happy to see it.
Yeah.
Quote, if successful, this approach could help make disease treatment simpler, potentially improve survival rates, and lower overall healthcare costs.
Okay, another positive there. Writing in Optica on February 12th, 2026, this year, Optica Publishing Group's Journal for High Impact Research, Zhang and colleagues reported that sensor detected lung cancer biomarkers in patient samples at
Attomolar.
What is that?
Attomolar.
Subatomolar levels. Okay, I have no idea what that is, but they're the scientists. That means it was able to generate a clear signal even when only a handful of molecules were present. Because the system is programmable, it could potentially be adapted to identify viruses, bacteria, environmental toxins, or biomarkers linked to conditions such as Alzheimer's disease. Okay, it's getting better.
Let's get AI. Put AI in this so that it can find, all these different cancer diseases inside of you. You know?
And Alzheimer's.
Yeah. Oh, Parkinson's too. For that,
Yep.
Let's just throw it in the common cold.
Yeah.
All right. Quote, for early diagnosis, this method holds promise for enabling simple blood screenings for lung cancer before a tumor might be visible on a CT scan, said Zhang. It could also help advance personalized treatment options by allowing doctors to monitor a patient's biomarker levels daily or weekly to assess drug efficacy rather than waiting months for imaging results.
Most current methods for detecting biomarkers require chemical amplification to boost tiny molecular signals, a process that can add time, complexity, and cost.
Yeah.
The researchers aimed to design a direct detection method that avoids these steps. Okay.
The new platform relies on SHG, a nonlinear optical effect in which incoming light is transformed into light with half the wavelength. In this system, SHD takes place on the surface of a two-dimensional semiconductor called molybdenum disulfide.
Molybdenum, that's a really rare metal, I believe.
Yeah.
To fine-tune the signal, the team used DNA tetrahedrons, which are small pyramid-shaped nanostructures built entirely from DNA, to position quantum dots at exact distances from the MOS, is that squared or MOS2? What is that? Some
Molybdenum sulfide, disulfide.
sulfite? Okay. Surface. These quantum dots intensify the local optical field, strengthening the SHG response.
CRISPR-Cas gene editing was then incorporated to recognize specific biomarkers. When the Cas12a protein identifies its target, it cuts the DNA strands anchoring the quantum dots. This action produces a measurable decrease in the SHG signal. Because the SHG technique generates very little background noise, the system can detect extremely low biomarker concentrations with high sensitivity.
Okay, quote here. Instead of viewing DNA only as a biological substance, we use it as programmable building blocks, allowing us to assemble the components of our sensor with nanometer level precision. That's according to Zhang. Going on with the quote, by combining optical nonlinear sensing, which effectively minimizes background noise, with an amplification-free design, our method offers a distinct balance of speed and precision, unquote.
That's good, yeah. To evaluate performance, the researchers focused on MER21, a microRNA linked to lung cancer. After confirming detection in a controlled buffer solution, they tested the sensor using human serum from lung cancer patients, mimicking real-world blood testing conditions.
Another quote, the sensor worked exceptionally well, showing that integrating optics, nanomaterials, and biology can be an effective strategy to optimize a device. That's, again, according to Zhang. Going on with the quote, the sensor was also highly specific, ignoring other similar RNA strands and detecting only the lung cancer target. Okay, that sounds pretty good.
Yeah, if this can be programmed, that's great. To pancreatic cancer, whatever,
Yeah.
breast cancer. All right. The next step is to shrink the optical system. I hope it's not the size of a building. That'd be crazy. The team hopes to develop a compact portable device suitable for bedside use, outpatient clinics, or remote areas with limited medical resources. We love it.
Yeah, this sounds really, really promising here. So go Zhang.
Yep. Do it.
Cool.
Root for the Chinese on this one.
Yep. Yeah, let's just hope it's legit. So,
yeah.
But it sounds very promising. So thanks for listening to us this morning.
Okay, we'll see you in the afternoon.
Take care.